My name is Sydney Thompson, and this is Evaluation of Mass Spectrometric Method for Identifying Protein-RNA Interactions. First, I would like to start with a statistic. Glioblastoma is one of the most deadly brain cancers to exist, with only 10% of people who are diagnosed surviving 5 years past said diagnosis. This is a very serious disease, and we would like to do something to help, obviously. What we are looking at is specifically RNA and how it's associated with cancers like glioblastoma. RNA is basically how your DNA interacts with the rest of the cell, how it communicates. By studying this, we can see what the cell does when glioblastoma occurs, because cancer is a cellular disease and is associated with cells that won't stop reproducing. So by looking at how the cell itself works, we get a better idea of how cancer works. Our group specifically, that my lab and my project is a part of, uses mass spectrometry in order to look at whether RNA is doing anything with certain proteins. Now, we're hoping to take a look at these and see if it can give us some ideas of how the disease works and any ideas of how to treat it. But before we can look at that, we need to test our methods first, and that's where I come in. I'm testing certain ways of refining the proteins that we'll be looking at, and we're starting with a model called bovine serum albumin. Bovine serum albumin is a protein found in basically cow blood, to be very simplistic, but what we're doing with it is we're cutting it with trypsin in order to cut it into 82 peptides. These are smaller fragments of the protein that'll be a lot easier to see in mass spectrometry. And we're treating it with two methods of basically purification, a guanidination reaction and ZipTip purification. And we're seeing if these purifications help us see it better in the mass spectrometer, which we're going to see right down below. So the mass spectrometer shows us the proteins basically and their masses. That's what each of these little spikes are. Now, we were hoping to find 3 of these spikes to be the 82 peptides we wanted, but in every test, and we've run many of them, we've only ever been able to get 2 at once. This was our most successful test. We've sometimes gotten different ones in these 2, but we've consistently gotten number 27 and number 36 in our peptide sequence, which are identified by those yellow arrows. So what's going wrong? We're not really sure. There's a lot of background noise, basically extra lines that don't really relate to what we're doing. And we haven't figured out what's causing that. We think it might be the concentration, but we have upped it and even doubled it. So we're not necessarily sure if that's it, but we are going to be looking at that in the future. And we're looking at the protocol to see if there are any ways that we can simplify steps or take a look at making things more efficient. So right now, this hasn't really worked out, but we are looking at ways to refine it in the future, which is awesome. And ultimately, that's pretty much where we're at right now. Hopefully, we'll be able to find out a little bit more in the future so that we can get a better look at these proteins and eventually be able to move on to learning more about how glioblastoma and proteins in the human body interact with our own RNA. But for now, I'd like to thank the chemistry department at UNCG for use of their facilities, Marquis Star for funding, and I would like to thank my awesome lab mates, teammates, and co-authors for everything you've done to help me get this done and for setting up this whole idea. This has been an awesome experience, and I look forward to learning more in the future. So thank you for viewing.